This series is about biochemistry and structural biology of nucleic acids, and it's from a course that I gave for postgraduate students in Warsaw, Poland. It is structured into several different parts. I start out from the question why nature uses the building blocks that it uses in nucleic acids. I then cover classical themes of nucleic acid structure. From there, I move on to the biochemistry of DNA modifications, either from damage or from modifications that are actually enzymatically catalyzed. In this section, I will talk about damage to nucleic acids and also about enzymatic modifications of nucleic acids. Speaking of DNA damage, it's also interesting to see what the vulnerabilities of natural nucleic acids are. And of course, this is a concern for RNA. Obviously, there's debate even about RNA damage, although I'm kind of skeptical. I still think that RNAs are too short-lived to be worth really repairing very thoroughly. You just degrade them and make new ones. But obviously for DNA, integrity and stability in the long term is a major issue. And for me, the eye-opener paper is actually one review article by Thomas Lindahl in Nature Science, where he basically lays out the chemical principles. And what you can see is that you can basically understand the entire field of DNA repair in terms of the structural and chemical biology of DNA that debates the vulnerabilities of nucleic acids. And it basically turns out that sure enough, nature knows about all of them. If you understand the chemistry well enough, you could have predicted the entire DNA repair field. And in a way, I would argue Thomas Lindahl, with this very classic, also very historic paper already, almost did that. So he was basically coming from the physical chemistry, from damaging DNA under conditions of high temperature, under conditions of strong artificial oxidative stress, and so on. And he basically looked how it responds and then predicted that to a lower level, these same processes should be going on in normal conditions, and there should be defenses against it. And basically, eventually, that earned Thomas Lindahl and others the Nobel Prize in biomedicine for his work on DNA repair. But it's really all driven by an incredibly conceptual program that I admire to no end, as you can hear. So one of the things that he discusses as well is that this means that the information storage in nucleic acids is finite. So there are natural damage processes with intrinsic rate constants that basically show, and this was already clear in Lindahl's time, that basically tens of thousands of years are okay for DNA in favorable conditions. But after 150 million years, so 1,000-fold more, basically you expect DNA to be completely destroyed by its intrinsic liabilities. So there is a field of human paleontology, like the Svante Pääbo stuff that you've all recently admired when he was awarded the Nobel Prize for this, actually is compatible with the chemistry of DNA. And Lindahl saw this decades ago, and so did Svante Pääbo and others. By contrast, the Jurassic Park thing remains the domain of Steven Spielberg. So it's simply against the physics. You will not revive the DNA from a Jurassic Park dinosaur. It's just against the physics of it. So where does DNA get damaged? And this is a figure from Lindahl's original paper, and it's basically showing that it's almost everywhere. Right? So the nucleobases are in danger, the ribose is in danger, It's basically all over the place that DNA can be damaged, and it can be damaged in very different ways. So the first thing to notice is that, of course, you care about vulnerabilities of the backbone. Right? Miscoding a single base is bad, but breaking strands and completely scrambling the information in the genome is a lot worse. So not getting strand breaks is important. And actually, the thing that many people are not aware is that the transition from RNA to DNA with the loss of the 2' hydroxyl group, which you have in RNA and you do not have in DNA, is actually nature's measure to stabilize the phosphodiester backbone and to basically make DNA a practical long-term storage polymer that allows for long pearls on a string information carriers that will not fall apart too much. And basically, why is the 2' hydroxyl group such a problem for an information storage polymer? And the short answer is, it is because of intramolecular catalysis. And what you see here is that the 2' hydroxyl actually can attack the phosphate in the backbone, and you get an intermediate that is basically very easily falling apart, and it can fall apart in two possible ways. One reconstitutes the original dinucleotide, and then everything is fine, but the other decay pathway actually cleaves the phosphodiester backbone. And therefore, 2' hydroxyl groups are bad for RNA, are bad for an information storage polymer, and therefore they are removed from RNA, and therefore DNA is the much better solution chosen by nature. Now, what are the vulnerabilities of nucleic acids otherwise? And many of them concern the nucleobases, right? And basically the main threats are alkylation. So you attach an alkyl group. So in the simplest case, a methyl group, but could be an ethyl group, could be longer alkanes from tobacco smoke, for example. So, or it could actually be a methyl group from the biological methyl donor, S-adenosylmethionine, which is also doing unspecific alkylation. So alkylation is a concern, oxidation is a concern, especially under conditions of oxidative stress. Deamination is a concern. And finally, also, loss of the glycosidic bond is a worry. And of course, the first three alter the base, but they don't lose the base. By contrast, once you leave a glycosidic bond, basically the base is altogether gone. And we'll see why this is very bad. So let's now look a little bit at the different types of damages. So, and I'll limit myself to the simplest case, to so-called aggressive SN1 reagents. So the most undiscerning alkylators. And basically, what do? Right? They largely leave carbons in peace on nucleobases, so you don't expect carbons to be methylated or alkylated by chemical agents. For the nitrogen, it strongly depends, and oxygens are vulnerable. And it depends has actually been my research interest for a while. So you basically have a collection of cases. The imidazole ring of the purine on adenine, it happens on the N1 predominantly, to some extent also on the N3. On cytosine, you have the N3 being methylated, and basically among the oxygens, the most vulnerable oxygen is actually the guanine 06. And there's actually a dedicated protein called MGMT that is used to undo this particular type of damage, sacrificing one protein just to repair one such damage. And then on the other hand, there are alkylations or methylations that you really don't get at all. And some of these are the ones that enzymes do. And C methylations or C alkylations you don't get. And for example, you would not with chemical reagents get any reasonable amounts of 6-methyladenine. It's simply chemically unfavorable. And of course, you could learn all of this. And actually, if you read the older cancer literature, then there are extensive reviews that basically present this as a ton of facts. But actually, when you think about the chemistry and look at my recent review with Guoliang Chu from Shanghai, you can actually see that you can rationalize this information in terms of the chemical classification of the nitrogen and oxygens in the nucleobases. And the short conclusion is that there's one type of nitrogens that are particularly vulnerable, and these are the endo-N+ nitrogens. And when you know this one little thing, you can basically know all of alkylation damage to DNA in one rule. Okay. Apart from alkylation damage, the biggest other concern for nucleobases alone is oxidation. And oxidation basically is a problem primarily for one reason. So among the bases, the one that is most easily oxidized is guanine. And the guanine oxidation actually generates a damaged DNA base called 8-oxoguanine. And 8-oxoguanine is terrible because it has a tendency to switch from the favorable anti-orientation to the syn. And why is this? This is because before you go to anti, because you don't want the 6-membered ring of the purine to be in conflict with the deoxyribose. But once you have the keto group in 8-oxoguanine, actually you have an even worse conflict in the anti-conformation with this oxo group. And then actually it's the lesser evil to flip to the unfavorable syn conformation. And once you do that, the base pairing properties of course change. You're no longer exposing the Watson-Crick edge as you intended to, but you're now actually exposing the Hookstein edge. And the best base pairing partner for guanine on the Hookstein edge is adenine. And now all of a sudden, you get what's called a transversion, so not a transition, so not a purine to purine or pyrimidine to pyrimidine mutation, but what you get is that a purine, a G, becomes a T, and on the counterstrand, of course, the C becomes an A. It's called a transversion, and it's thought to be more severe than a transition mutation. Okay, there's also the issue of deamination, hydrolytic deamination. And in DNA, actually, the nice thing is that as long as you only work with the 4 standard bases, actually you create bases that shouldn't be in DNA in the first place. Basically, if you deaminate cytosine, so you hydrolyze cytosine at the C6, you get uracil, and of course, nature knows that uracil is meant to be in RNA, but it's not meant to be in DNA. So it's clear what is wrong, and it's clear how to repair it. Same for adenine. If you deaminate adenine, you get inosine, and inosine, of course, is not guanine. So nature knows it's fully in DNA, it shouldn't be there, and it's easy to repair it. And all the hazard starts once you methylate. And methylation, of course, you do for epigenetics, so it gives another layer of functionality to nucleic acid, but you pay a price, and the price is that now you subvert deamination repair. Because once you deaminate, you hydrolyze 5-methylcytosine, you get thymine. And initially it's paired with the guanine base pair partner of methylcytosine. So if you catch it early, it's repairable. But after one round of DNA replication, actually the change is sort of forever, because now it looks perfectly unsuspicious. And basically you can see that methylation actually cytosines that are frequently methylated tend to disappear from genomes, right? And I'm aware of this from own work some 10 years ago with Marek and Honorata, primarily in the group, where we showed that this is true for bacterial genomes, that basically you can find CpG-specific methyltransferases by looking for extreme underrepresentation of the CpGs. And the same is, of course, known in eukaryotes. Invertebrates mostly don't methylate their DNA, or they methylate it only very sparsely in a CpG context. And therefore, you have almost as many CpGs as you would expect based on the CG content of the genome. By contrast, in the vertebrates, actually 80% of the CpGs that we should be having in the absence of deamination repair actually are gone. And only some 20% of our CpGs are left. And as the species evolves, we are actually in danger of losing more of that, right? And of course, there's counter selection by the functional advantage that CpG methylation provides, but we've already gone a long way towards damaging our quote unquote original epigenome system. Finally, glycosidic bond cleavage. So glycosidic bond cleavage means that we lose the bond between the base and the deoxyribose. And that's actually bad, and this is to show you why it's bad. It's bad because once you lose the base, actually you're very much in danger of breaking the backbone of the DNA in addition. And actually what happens is that with the loss of the base, which is hydrolytic, you basically get the possibility for a ring open form of your deoxyribose. And basically the ring open form is prone to beta elimination, either in very basic conditions or under shift-based catalysis with primary amines. And basically what happens is that by beta elimination, you basically release the free primed end of the DNA. And technically there are two variants of it called beta and delta elimination, not relevant for this lecture, but what you should remember is that losing the base is bad because next you will also break the backbone. And of course then the single strand break is a lot more severe than the original problem. Note also that the beta elimination that takes place involves the 2' hydrogen atom in the 2' deoxyribose. So basically, and this is a point very nicely made by Thomas Lindahl when you read his review, basically the transition from RNA to DNA very much stabilized the backbone against the intramolecular, the catalyzed cleavage via the cyclic intermediates that I've been showing you in the beginning. So it was a good defense against unwanted hydrolysis of the DNA backbone. But at the same time, it has created a new vulnerability in that base loss is now really severe and tends to generate DNA strand cleavage. But apparently, this is the lesser problem than the direct attack, because here you only need to stabilize the few bases that have the few nucleosides, or 2' deoxynucleosides, that have lost their base, whereas in the other case, you need to stabilize all of them. So basically, it's a good idea for overall stability to pay this price in order to solve the hydrolysis of the backbone problem. You can then live with the elimination. Okay, finally, enzymatic modifications of nucleic acids. So in DNA, actually, the possibilities are very limited. And they're very limited because there are lots of things that the modifications cannot do or else be unacceptable. And obviously, they should not alter what's script pairing because it mess up heredity. they should not interfere with DNA replication and transcription. So you don't want to modify, say, the outer minor groove positions in double-stranded DNA. So say the N3 of adenine, even though it would be facile, because that's where the polymerases need to grab the nucleotide. And of course, you don't want to use anything that destabilizes glycosidic bonds. No electron withdrawing groups are tolerable, because if you had them, you tend to lose bases. And if you lose bases, then you're one step away from basically making the DNA fall apart. So with all these constraints, you can actually show that basically of the alkylations or methylations that are possible, basically the three main nature's choices, 5-methylcytosine, 4-methylcytosine, and 6-methyladenine, are actually almost the only ones that are possible. Everything else runs into trouble. And you can really enumerate almost what that trouble would be. Really go for it design-wise and see what you would get. And you can argue them out one by one, why they shouldn't be there. And actually, it also means that the enzymatic methylations all end up being chemically difficult methylations. So it's the ones that would not occur naturally. So you would not methylate a C5 of a cytosine chemically. We've said that chemical methylation leaves carbons alone. Likewise, you would not methylate an exocyclic amino group. Qualitatively, why? Because the lone pair is conjugated to the aromatic ring. And when the lone pair is conjugated to the aromatic ring, basically you withdraw electrons, and then the nucleophilic attack on the methyl group in SAM is bad. So it's not going to happen. So enzymatic methylation is difficult methylation. And in part, that's because most of the easy methylations would not be tolerated. But it's also a good thing from a different angle. It basically means that you very cleanly distinguish between epigenetic signals and the one hand and DNA damage. And no DNA damage will mess up epigenomics, because basically they just target different sites. Okay, the 4-methylcytosine is a bit a niche DNA modification in the sense that it's almost exclusively found in hyperthermophiles, and that's because especially 5-methyl-C becomes problematic in hyperthermophiles for chemical reasons. But 4-methyl-C is otherwise not very favorable because it makes replication problems. So for organisms that don't live in conditions of very high temperature, actually the focus is on 5-methyl-C and 6-methyladenine. And the thing to notice here is that structurally the two modifications are very different. So first of all, in the favored context, dinucleotide context, so CpG and APT for cytosine and adenine methylation, the cytosine methyl groups are far apart from each other. And they actually nicely integrate into the stack of DNA base pairs, so that what you get is you stabilize dsDNA. And I tend to think that over evolution, this was the reason why 5-MC has become a repressive mark. I would say it's become a repressive mark because the stabilization of double-stranded DNA is actually biasing against transcription. And then, of course, the bias is way too small to suffice alone to explain the repressive effect, but it sort of gives a starting point for evolution to play with, to then basically develop all the epigenomic machinery that represses transcription in modern day organisms. 6-methyladenine, the exact opposite. So the 6-methyladenine groups actually clash with each other in the APT context. And when I was initially preaching this at conferences, people were laughing at me and saying, come on, that's ridiculous. The DNA base is stack, so the methyl groups will. Right? So how can you possibly say that methyl groups in bDNA are in steric conflict? It has to be nonsense. And the answer is it's not nonsense, and it's not nonsense because of propeller twist. So bases are twisted, and because bases are twisted, actually the twist would drive the methyl groups into each other. And basically the presence of the methyl groups enforces distortions of the DNA, and at the same time, it actually stabilizes this deformed conformation, and it protects DNA very much against certain other deformations, as we will see. And basically, if you look what 6MA does as an epigenomic signal, then there's very good evidence now in primitive eukaryotes, particularly algae, green algae, and the like, that 6MA is actually a strong activatory epigenomic mark. There's also a big literature on 6MA being a repressive mark in higher eukaryotes, but it doesn't make any sense biophysically. And basically, it's now, in my eyes, pretty clear that the papers that claim that, including all the higher profile papers, are basically completely implausible biophysically. They are done with antibodies, and at the abundances of 6MA in higher eukaryotes, this will be the best antibody that the world has ever seen. So it's just not believable. So basically, 6MA is a destabilizing modification biophysically, and therefore, in organisms that use 6MA epigenomically, it's an activating mark. 6MA, for reasons that I don't have time to go into much, is a moderate problem for replication. So therefore, higher eukaryotes see it as a form of DNA damage and avoid it. Now, an interesting thing is also the stabilization. So once you adenine methylate the APT context, basically you block against certain DNA deformations. And when you basically model DNA that has been wrapped on nucleosomes, you see that TPA methylation would be okay for the wrapped DNA structure that you find in nucleosomes, but APT methylation is completely incompatible. So if you wrap the DNA on nucleosomes, you would drive the methyl groups even further into each other than they would already be driven into each other in bDNA. And that explains why 6MA is very strongly anti-correlated with nucleosomes. That is clearly confirmed biochemically, it was not structurally explained. The structural explanation is ours. And it basically also explains on another level why 6MA is an activatory mark. It's an activatory mark because it destabilizes double-stranded DNA, which is good for transcription, but it's also an activatory mark because it's mutually incompatible with nucleosomes. And of course, getting rid of nucleosomes is good when you want to start transcription. Now, when it comes to nucleic acid modifications, the common ones sort of in lower and higher eukaryotes are 6MA and 5MC, comes out that there are a lot of lesser well-known ones, and most of them simply play a role in the warfare of prokaryotes against phages and phages against prokaryotes. And particularly the phages, who are very inventive in how they could decorate their DNA to basically become resistant to the attack from the host by endonucleases. Of course, the phages still need to respect that their DNA has to remain an information coding polymer. So the choice of anchor points of modifications is very restricted. But then what you put on there, then you really have a lot of freedom what you do, and phages use it to the full. Now, when you think about modifications in nucleic acids that are RNA, then a lot of the arguments for what constrains them go away. So in RNA, actually, there's no issue with replicating the RNA again. So if you do it post-transcription, then you're not worried about replication, because anyway, you have lots of non-Watson-Crick base pairs. You don't care about messing up Watson-Crick base pairing. And so lots of constraints go away. And because lots of constraints go away, you get an immense plethora of possibilities. And this is actually just a snapshot of what the different lineages of life actually invented to do for modifications, right? And they do all sorts of things, right? They stabilize structures, both canonical and non-canonical, Watson-Crick pairing structures when it's wanted or not wanted. They modulate the binding properties of the anticodon loop. So lots of stuff that you can do with RNA modifications. And this is a very, very rich field. And if you want to see how rich, then look at Janusz and his group's Modomics, huge database that actually tells you how creative nature has been in what one does with nucleic acids. So in summary, this is Tile in Notre Dame University in Indiana with a famous quote from Theodosius Dobzhansky, the big evolutionary biologist, who is known for saying that nothing in biology makes sense except in the light of evolution. And I hope the spirit of this talk convinces you that one could also say that nothing in biology makes sense except in the light of structural chemistry, except certainly when it comes to nucleic acid chemistry. And with that, thanks for listening, and I'm happy to take questions. You see the chat. Does anyone want to ask questions? I see the question. Is it possible to allow also other attendees for recording? Just the ones of us that have to do the exam and need for studying well in advance because the videos are made available. So maybe we can solve this in the way I'm told that I'm allowed to. I will get the video early, and I was actually anyway planning to put them on YouTube, to open a YouTube channel and put them on YouTube. So if Ivona, I don't know, Ivona, are you here? If Ivona allows, like last time, and if I don't find any embarrassing mistakes in my delivery of the lecture, then I would just put it on YouTube, and that solves the issue.
Am I audible?
I can't hear you. Can you repeat that, please?
Yeah, you can hear me, right?
Yes, now I hear you. Yes.
Okay. I had a question. Like, what about like complex 4D structures of the DNA? Like, when the DNA based on their grooves and form like tertiary structures, not like
I really struggle to understand you. Can you speak louder, please? I barely hear you.
Is it better now?
It's better, yes.
I was thinking, like, how does this like translation and everything affects when the DNA takes on more complex structures, like 3D structures, not like tertiary structures, I mean.
But actually, DNA, I mean, there are DNAs that take on tertiary structures called aptamers, DNA aptamers. But in general, actually, the dogma is that DNA is not supposed to take tertiary structures, right? And it's actually one of the benefits, apart from the increased stability. If you take out the 2' hydroxyls from the ribose, actually, you take away a lot of the interactions that stabilize tertiary structure in ribozymes. So DNA is not meant to make too many non-canonical structures, right? Artificially, yes, but naturally, my take is it's not what nature is about. So very little there, right? And you talk about translation, I didn't get that point at all, right? DNA is far away from translation. Translation is in the cytoplasm, and the RNA is transcribed in the nucleus and then exported. So I missed your point on the DNA.
I meant transcription, like DNA to RNA. I just misspoke, sorry.
Okay, yeah, sure. No, but again, for transcription, right, the major interest is that the DNA represents a sort of universal phase, right? That basically an RNA polymerase would basically be able to do the same thing at every step, no matter what the base sequence is, right? So if you have these structures in DNA that tend to form non-canonical structures, right, it's a problem for replication, right? So I would actually argue that biological organisms are actually optimized to avoid that. And you don't want stuff where a polymerase would struggle because the DNA structure looks non-canonical.
That makes sense. Yeah. Thank you. Thank you.
Thanks. Any other questions or comments? I don't think I see. Am I missing? Speak up if I miss anyone, but I don't see any comments on the chat. So I think then we are done, right? Thanks again for attention. I will try to get the recording from Ivona Pilecka, and if I can get it, I will put it on YouTube together with the previous lecture. Okay? Okay, thanks. Bye-bye.